meta-analysis in Jamovi and drawing a forest plot and funnel plot. Meta-analysis is a systematic review, literature review, and statistical method that combine and summarizes the results from several independent studies which investigate the same research question. So the analysis will provide precise estimate of the effect size of exposure or treatment or experimental compared to a control if the setting for that research was in that format. And the meta-analysis will pool all the results from multiple studies and then will generate an overall estimate effect size. The meta-analysis will analyze data from previous already research studies that has been done and pull them together to identify any common patterns and make a conclusion about the research study and whether there is a difference or an effect between the compared two groups, such as experimental or treatment or exposure to control. The meta-analysis will measure the variability of the study results. There is any variability between the each research study from another, and calculate the confidence interval for each study and perform an I-square test. I-square test or homogeneity test, or sometimes it's called heterogeneity test, is a test that will measure the degree of variability or inconsistency in the result among the multiple studies in meta-analysis. The value for I-square is expressed in percentage. An I-square value with 0% indicates no heterogeneity, and an I-square value of 100% means maximum heterogeneity. The meta-analysis will also generate a forest plot. Forest plot is a graph that represents the results from meta-analysis with summary attached. The plot consists of three major parts. The first part is a statistical analysis and statistical description for each study with the number of participants and the estimate size. The second part is the diagrammatic representation of the data output, data analysis output, statistical analysis, and the third section is the overall estimate of the effect size and the p-value and the test of homogeneity or heterogeneity. Another plot that can be generated from meta-analysis is called the funnel plot. It is a graphical display of the effect size estimate plotted against the standard error or the sample size. The plot will assess the publication bias in the meta-analysis. To do meta-analysis in Jamovi, here I had inserted data from 10 independent studies addressing a specific question. In the first column, the study numbered from study number 1 to study number 10, and the year of publication is given between two brackets. And the next column is the number of participants. So this research study has been designed to have two groups: the treatment or experimental group and the control group. So this column is the number of participants in the treatment group, and then the mean for the treatment group is provided here, and the standard deviation for the treatment group. And for the control group, the number of participants is recorded, and the mean for the control group and the standard deviation is also scored and inserted here in these columns. So here we have an outcome which is continuous, and the means, one of the parameter for that continuous variable is the mean. And there are certain options that we can select to compare and do statistical analysis in those combined studies, either looking at the mean or the standardized mean difference, or using an effect size such as Hedge's G. To do the meta-analysis in Jamovi, you need the function for that, and if it is not installed meta-analysis function, then click on the modules here, and then click on Jamovi and select the meta-analysis options from here, and then install that. Once it is installed, click on the major and scroll down. Since our data here, it is not correlation, it is not binary outcome, it is size effect, and we will select the mean difference, as we have the number, the mean, the standard deviation, and then we could add more modification subsequently during the analysis. If we click on that, a new window appear here, and for the mean differences, and we need to move each specific variable or data into the right section. So the study, or the name of the studies, or the list of the studies goes for the study labels, and then for the group number 1 and group number 2. So we have here two sections, so we'll move the number of cases to the first box, the mean for the group 1, and then the standard deviation. So these all the data for the first group. For the second group, which is the control, move the number of cases to the group 2, sample size, then the mean to the group 2, and the standard deviation for the control for the group 2. If we stick to the estimate effect size, which is the mean difference, two tables are generated, and the model is selected as a random model, and we have 10 studies here, k equal 10. And the important one, which is the effect size, which is the estimate, is 0.73. This is the overall effect size, and it is significant as it has a p-value of less than 0.001, and the confidence, 95% confidence interval, is for the lower limit and for the upper limit, and the estimate lays between these two limits. So there is a statistically significant difference between the treatment group compared to control, as the overall estimate effect size is 0.7. Looking at the heterogeneity between the studies, a test has been done, which is the I-square value is generated, and the value is 20.1%, which means that there is minimum heterogeneity between the studies. We need the p-value to be greater than 0.05, and here it is 0.3. So there is to a certain level minimum heterogeneity between the studies. If we scroll down here into these options that are available, we could also select some of the options. For the model estimator, we could go for the Hedges G and select that, and that will also generate a few modifications. So we could stick to the one which is for the Hedges G value, and for effect size model measures we'll keep it standardized mean differences, and no changes for the moderator type. Confidence interval 95%. Display model fits, no need for that. Let's go and take on the plots. So we need to draw now two plots, the forest plot and the funnel plot. And if we scroll down here, we'll see that a forest plot is generated, and on the left hand section is the studies from 1 to 10 with the year of publication, and the line, dotted line here, is the line of no effect, which is 0, and the square symbol here represent the in this case the effect size, which is the Hedges G, or the standardized mean difference, whichever option you prefer to go for. And as shown here, each square have different sizes, and that size is larger square, small square. That size of the square, which is the effect size, reflect the sample size. Larger sample size will have larger square, and it will have higher weights percentage influence in the analysis. The horizontal line here for each study represent the 95% confidence interval, and the diamond shape is the overall estimate effect size, which is in this case is given as 0.72. Looking at the right section here, each study will have the effect size, which is in this case Hedges G. For every individual study is calculated with the confidence interval. From the forest plot, the sections are here given for the diagrammatic representation, the statistical description, and for homogeneity test is given in a separate table. Here for the forest plot, no major changes will be done, only if the interest in changing the sizes and colors of the effect size point, or the study order, or whether the forest plot is size is small or big, or even I can go and change the effect size points into, let's say, empty squares, and this will reproduce the graph with empty square. Moving to the funnel plot, which is the last section here, as shown in the funnel plot, the plot is have an y-axis and x-axis. In the y-axis is the standardized error, and on the x-axis is the standardized mean differences. And I could go for selecting for the y-axis either the standardized error or the sample size, whichever you find suitable. I will stick with the standardized error on the y-axis, and on the x-axis is the standardized mean difference. I could also select the plot size, whether medium, small, or large. The funnel plot will assess the publication bias. As shown here from the plot, the middle line is the overall effect size, which is 0.72, and the small circle here represent the effect size for each individual study. The effect size study should be evenly distributed above the line for the overall Hedge size effect, and the lower part. So the dot here should be evenly distributed in these two sections. Here we have 1, 2, 3, 4, 5, and here we have 1, 2, 3, 4, and there should be no dot outside the funnel plot. So this funnel plot will reflect whether there is a bias or no bias in the publication. So this is the way to do meta-analysis in Jamovi, and drawing a forest plot and a funnel plot